I will give you a brief overview of current knowledge and insights in liquid biopsies in cancer, which is a quite broad domain. I realize that when I look at recent papers in relation to this topic, but I hope I can summarize the main concepts of it. Okay, liquid biopsies, an attractive concept that you can do examination of samples of your patients by just taking a blood sample instead of doing a biopsy or a bone marrow aspirate, as it has to be done often for patients with hematological malignancies also. And therefore very attractive, and of course a lot of efforts are currently done to examine the value of those liquid biopsies. Of course, it's not a new concept because already many decades ago, people tried to use blood as a tool to monitor cancer by looking at tumor markers like PSA or another antigen or marker that's related to ovarian cancer, for example. But although these markers do have offer their benefit overall, there are some problems with specificity, and also the sensitivity is not always high enough to do a careful monitoring of the disease. Certainly when you want to define deeper remissions. So that's why more attention is given now to let's call them new biomarkers, including the circulating tumor cells, circulating tumor DNA. More recently, studies are started up to examine the potential of circulating exosomes, and there are also, of course, a lot of studies dealing with microRNAs in blood. I will mostly focus on these three, both in the introduction part as well as in the second part, dealing with our own project. Now, why is it so attractive to have a good test that you can perform on blood samples? It can allow you to have an elegant test to do an early diagnosis of the disease. It can help you to select the most appropriate therapy. It can help you to give an indication about the prognosis, and during treatment, it's a tool that you can use to do therapy monitoring. So what are the specific advantages? There are no risks and side effects which are normally linked to biopsy collection and marrow aspiration. And I know that this might not be overestimated, but it can sometimes be a problem in some patients and in some indications. It allows you to have a better assessment of the overall tumor heterogeneity, because when you do a local biopsy collection, or even in a disease like myeloma, you do a marrow aspiration, you have, of course, a very local part of the tumor mass that you do examine, while in the blood you would expect to have a more general reflection of the overall tumor mass. It allows you evolution in real time, and as such, it is also possible to do more frequent sampling. Taking a blood sample is quite simple. You can, as such, have a more accurate disease monitoring and therapy evaluation. And overall, it might also help you to change your therapeutical attitude when you find out quite early in the treatment that a tumor marker is rising in the blood. Now, that the topic is quite exciting and attracts a lot of attention from research teams, is reflected by these curves. In fact, it's a review article in Oncology where the author tried to count all the publications that were, in fact, that are visible on PubMed in relation to those specific keywords. And you see that when we do look at liquid biopsies, that's the pink curve, that topic is increasingly appearing on PubMed over the last decades, 2-3 decades. When you do look at the circulating tumor cells, in fact, that curve is quite going in parallel with the first curve. And then you see some curves that are rising more recently, the ones that deals with exosomes, and here the circulating tumor DNA. Here you have the highest curves dealing with microRNA and cell-free DNA, and this starts to rise much earlier or somewhat earlier, but this includes also studies that are not related to cancer. That explains why there is such a high incidence. The drop that you see here is just due to the fact that the follow-up was stopped in the middle of 2017, so 2017 is not completely included in the curve. Now, let's start with the circulating tumor cells. These cells are assumed to be, at least part of them, to be responsible for disease spreading and metastasis formation. It are, of course, rare cells. Their number is estimated to be around 1 out of 1 million or 10 million white blood cells. And to those who succeed to isolate those cells, it is assumed that normally there should be in cancer patients around 10 of these, between 1 and 10 of these CTCs per 10 ml peripheral blood. That's a rough estimation, because in some indications it can be much higher, and in other patients it's not detectable at all. What is known about those CTCs is that they can, at least part of these cells, can be involved in a process called epithelial to mesenchymal transition, and that changes the features of those tumor cells in that sense that the phenotype, for example, can become different, and it can cause a problem in terms of isolating those cells, because sometimes they lose an essential target antigen which is used to capture these cells. They can be found as single cells, but sometimes also as clusters with other cells that can be accessory cells that do support, in fact, the survival of these cells. And it turns out that, in fact, the tumor cells in those microclusters are more viable than the single circulating cells. Most single circulating tumor cells, by the way, will not survive and will die off by apoptosis. What is the biological fate and significance of these CTCs is not always clear, and it is definitely not shown for all tumor models that they have indeed a pronounced metastatic potential. So functional assays are sometimes lacking there. Now, in terms of isolation, as I've told you, this isolation is mostly based on antigen expression. A lot of studies dealt with have looked at the selection of EpCAM positive tumor cells, which is a main epithelial cancer cell antigen, combined sometimes with depletion of CD45 positive cells, which are white blood cells, and there are some studies indicating that EpCAM can be weakly expressed on some white blood cells as well. Therefore, some groups do go for a double isolation step, depletion of white blood cells and isolation of the EpCAM positive cells. A problem, of course, is that at least a part, and in some tumors more than a part of the circulating tumor cells can become EpCAM negative because of that transition to mesenchymal features. And therefore, alternative methods of isolation have been looked for based on physical properties of the cells, cell size, density, electric charge, and so on. So lots of prototypes of devices have been used and developed so far, but there is clearly, with one big exception, lack of multicenter validation, and the reproducibility and also specificity of these approaches are quite unclear for the moment. Moreover, what is a major drawback in this whole approach is that there are no standardized protocols for the sample processing, the storage, and the enumeration. Now, once that those cells are purified, suppose that you can isolate enough of these cells, you can use them for different purposes. You can just count them and try to relate their number with some characteristics of the disease. You can use them for molecular characterization. You can use them for in vitro drug testing, at least if you can purify enough of them. You can try to derivate cell lines from them, as some groups have done, and you can use those cell lines to further characterize the biological features of circulating tumor cells as such. About those molecular characterizations, one can look in particular at the DNA that you can isolate from these cells and look for driver mutations for particular diseases. You can look for drug resistance related mutations. You can look for specific tumor markers in the DNA, as we do for B-cell malignancies. You can also use the DNA from the CTCs for methylation analysis. You can look for translocations. And you also can isolate RNA, look at the gene profiling, and also proteomics. That's all theory, but fortunately, a part of these aims have been achieved in experimental studies. Now, I told you there is one reference system so far in isolating circulating tumor cells. It's the self-search device, also known as the Veridex, that is already for a while on the market, which is FDA approved, and in the States as such approved for at least enumeration of CTCs in a few specified indications. It's also based on immunomagnetic cell selection, based on anti-EpCAM antibodies, which are coated to magnetic particles. And in fact, the whole platform exists, is composed of two devices. You have, first of all, the isolation device, where you can isolate the small amounts of tumor cells. And then you can use those cells to label them with antibodies. One antibody that does recognize your epithelial cells by means of cytokeratin staining. An antibody against CD45, which is a marker that is used to estimate the contamination with white blood cells. And then a stain which is used to stain the nuclear part of the cell, so a nuclear dye. So it's a nuclear staining. By using these three markers, cells are labeled, and then in this kind of semi-automatic fluorescence microscope, you can count the number of cells. It's a device that scans, in fact, in a chamber all your positive cells, and from each positive cell, a signal is sent towards a screen, and you have always there's always an operator needed, so it's not a completely automatic system, and that operator has to look at the screen and has to judge whether it's a CTC or not. The phenotype is helping you a lot, of course, but also the morphology is taken into account, and there are justified criteria for that, but you need trained technicians who look at those cells and who do the counting. Now, certainly in metastatic breast cancer, a lot of studies have been performed with this device. That's also the reason, because indeed correlation with the clinic has been shown that the device was approved by the FDA, not only for breast cancer, by the way, but apparently also for prostate and colorectal cancer. And indeed, referring to the very first study where the prototype of this device was used, it was shown that when you make a cutoff of 5 cells, positive cells in the blood, and they start the analysis also always with a tube of about 7.5 ml. So when you are having more than those 5 cells in your analysis, you belong as a patient to a worse category, because both the progression-free survival and the overall survival is in fact lower for those patients who have 5 or more of these cells compared to the patients who have less than 5 of these cells. Now, this study, or this type of studies, have been performed later on with other types of malignancies, and also for other cancers. Cutoff values have been defined for colon cancer, for example. The cutoff there is 3, which is of course quite low. And then other malignancies like lung cancer and renal cell cancer, also there a lot of studies have been performed, but with less convincing and sometimes conflicting outcome. For lung cancer, especially the non-small cell lung cancer patients, seems to be a problem that you have a quite high percentage of having no detectable tumor cells present, and so making their correlations with survival is and outcome is sometimes difficult. Now, limitations are certainly linked to the fact that on certain tumors the EpCAM expression is less strongly and less frequently observed. Efforts were done to develop other devices, because you cannot explain it all by looking at the phenotype. It might be the technology of the isolation device as such that also, of course, influences. Some years ago, there was a company coming along that tried to convince us that the ISOFLUX device was a better alternative as the cell search device, although it was also based on EpCAM expression. But they did a comparative study in lung cancer patients, and they claimed, you see the orange bars, which indicates the number of tumor cells counted when they used the ISOFLUX device, and the blue ones with the Veridix cell search device, and you see that there were much more patients where this device was able to pick up tumor cells. It was the story how it was presented when they came over here. Now we had the opportunity to test the device here, and when you work with a tumor line and you do spike experiments, you can indeed conclude that you succeed to isolate quite low numbers of tumor cells with a line. But when it comes up to patient samples, we were in fact less successful in that sense that you can isolate small number of cells, but we were on a group of 10 samples in fact not able to demonstrate that the mutation that was found in biopsy material and at the moment of diagnosis, that this could also be shown in the isolated tumor cells, and therefore the interest and the optimism was somewhat lost for this device. And at the end, you cannot ignore that the EpCAM negativity is also here a crucial problem, because it's not because it is not a device that it will change anything at the targeting as such of your cell at the antigen level. Another limitation is, of course, also the sample volume, because most of those devices were mentioned to isolate cells from samples of about 7.5 to 10 milliliter. But you can argue when you use more sample volume, you should theoretically be able to isolate more tumor cells. And a device that has been proposed quite recently is a quite simple one. It's in fact some kind of in vivo tumor cell selection. It's this type of device. It's in fact a type of catheter, a cannula, which is injected into the arm, and then you inject in that cannula a very small weir, and that's coated with hydrogel in which antibodies against EpCAM are in fact present. And by leaving that cannula for 30 minutes in the arm of your patient, you have some kind of in vivo enrichment of your tumor cells. This has been done apparently in one published study for lung cancer patients. It was a heterogeneous population, but most of them were non-small cell lung cancer patients, and the authors claimed that they had a detection rate with this system of 58%, which was quite high, and they claimed that in comparison with the cell search, they could only detect in 28% of the cases positive tumor cells. So they were quite optimistic about the possibilities of this device, but I think we have to see how this will now further be evaluated by other groups. So far, they only have one type of wear available, and that's the one coated with anti-EpCAM. No other antibodies or targeting of other tumor cells is possible so far. Now, another possibility is doing to collect more blood is definitely the case in this situation where you do a diagnostic leukapheresis. You can process with a leukapheresis device 50 liters of blood. We are familiar with this application to collect stem cells, as we do it in that way. And as published in this publication, in fact, what they have done, they have evaluated the approach in patients with non-metastatic breast cancer. And I look at the article because I was curious to see how much blood that they treated on the collection device, but apparently it were frozen stem cell products and stored stem cell products that were not longer used for transplantation purposes, and they did it on thawed cells. So they used the thawed cells, they used those thawed cells in combination with an adapted version of the cell search device, and they claim that they could detect with the cell search principle, if you want, or technology, in more than 90% of the cases, circulating tumor cells in the leukapheresis products. But I think looking forward and doing this in a prospective setting, it's not evident to ask your patient to come to be connected with such a collection device for 4 or 5 hours just for preparing a diagnostic test. Now, circulating tumor DNA is a more recently explored possibility. It is known that tumor cells, as well as normal white blood cells, do release DNA in the circulation. Not only in the blood circulation, by the way, but you can find cell-free DNA also in urine and other body fluids. It is estimated that in a healthy donor, up to 90% of that cell-free DNA is indeed coming from white blood cells. And in cancer patients, part of that cell-free DNA is, of course, tumor-derived, and we call that part, of course, circulating tumor DNA. It is estimated that 0.01% of your cell-free DNA is real tumor DNA, although this might vary from one patient to another. The release mechanisms of cell-free DNA are not so much known about. It is believed that the most important mechanism should be the fact that cells release DNA after going into apoptosis. There might also be a release of DNA after necrosis of the cells, and there are some groups who believe that there should also exist active DNA secretion mechanisms, but there's not so much known about it. The half-life of cell-free DNA is about 2 hours, looking mostly at fetal cell-free DNA. It seems to be that this disappears in 2 phases, that one part has a half-life of 1 hour and the second of 13 hours, but overall it has a quite short half-life, certainly compared with other biomarkers. Rise of cell-free DNA in cancer patients has been observed, but we have to be careful to relate this with tumor mass and tumor burden, because in cancer patients cell-free DNA can increase because of other released by other cells. Immune-competent cells, cancer accessory cells can also release cell-free DNA, certainly when you have inflammation going on in your patient. So the absolute amount of cell-free DNA is not so clearly related with your tumor mass. However, when you do efforts to look at tumor DNA by looking at that isolated DNA more based on tumor-specific markers, you can better correlate those levels to tumor burden and also therapy response, as shown in various studies. Now, a few considerations about the technical aspects, because I assume that many of us are interested to look at cell-free DNA, but it starts already at the moment of collection of your sample to have a good outcome of this type of assay. The question is, do you have to collect plasma versus serum? There are studies indicating that the concentration of cell-free DNA in serum is higher than plasma, but it is believed that this is mostly due to the hemolysis process, and that part of your white blood cells might also release DNA, and that it's not really reflecting the cell-free DNA you're interested in. So the preference is given to plasma, in fact. The collection tubes are also important. You can use EDTA tubes, but then you should start the processing already within a few hours. When you want to collect your tubes for a longer period, preference is given to vacuum tubes and or tubes that give a fixation to your cells so that they cannot release DNA in the tube before you can use them. Centrifugation steps are essential because you want to get rid of all your cell material, of course, and storage at -80 degrees is important as well. Companies have provided very easy to use kits, dedicated DNA extraction kits that can be used for this kind of purposes. And then when it comes to the analytical parts, as I've told you, you can use DNA in circulation, whether it's coming from cells or in a cell-free form, to do DNA sequencing. You can use PCR techniques, you can use next generation sequencing, depending on the depth of sequencing you want to achieve. You can look genome-wide, which is, of course, not so an elegant way to do when you want to have an easy-to-use diagnostic test, but you can also go for a more directed sequencing of a well-defined panel of genes, and you can also use that DNA for DNA methylation analysis. So the targets are here, as already mentioned, for the CTCs, also driver mutations, drug resistance related mutations, and then the other features that can be found in the DNA as well. Now, what about clinical relevance and limitations? There are several review articles dealing with this topic. I just mentioned one of the most recent ones that summarizes a lot of information and studies. I will keep it briefly here to the most important conclusions. Cell-free DNA as such, certainly when you quantify that, doesn't seem to be a good marker in that sense that the specificity is quite low. On the other hand, when you do efforts to really measure circulating tumor DNA, that seems to be to have more potential as a circulating biomarker, and in those studies where they do relate do compare the mutation profile, for example, in circulating free DNA with tumor-derived DNA, they find often a very good concordance in frequencies between tumor cells from the tissue or cell-free DNA. Now, ongoing studies do evaluate on a large scale the potential of that circulating tumor DNA for early diagnosis, prognosis estimation, monitoring of treatment efficiency, and minimal residual disease testing, as well as the identification of resistance mutations. That this whole approach is going into the good direction might maybe be reflected by the fact that FDA approved already some mutation detection tests. One to pick up specific mutations in the IGFR, which has its importance in selection of patients for clinical trials, patients with non-small cell lung cancer. But it's a specific test for cell-free DNA, which is approved now, and more tests might be approved soon. Also here, you see in several papers that there should be more validation and standardization, certainly of the pre-analytical methods, how the samples are collected and stored, can have its influence when those samples are used to do multi-center studies, so that's something where people have to work on. And then there remains also the observation that you sometimes fail to find circulating tumor DNA in patients where you could expect it to find, and it's clear that you have to be careful with that kind of patients, that you have here clearly limitations to use that parameter as a prognostic factor, and that you there definitely have to look always at information that you got from tumor biopsies. And then the exosomes, which is the more experimental part, I think. Exosomes are small membrane vesicles that are released from diverse cell types. They do circulate, they are released from cells, and they carry different types of functional biomolecules, DNA, RNA, proteins, and lipids. They are believed to be important in intercellular communication and play a role in cancer development and progression. To have an idea about the concentration that can be in cancer patients be above 10 to the 9th vesicles per ml. So also here, commercial kits, or at least one that we know is available now to isolate in a quite easy and short way exosomes from plasma samples. It is believed that the exosomes do contain longer DNA fragments, which can be an advantage for PCR assays. And the RNAs that are in there, the microRNAs, are of course better protected from RNase activity, which might be an advantage as well. Now here,
exosomes are not necessarily cancer derived.
No.
Normal tissue derived also.
They can be normal tissue derived as well, because in some of those studies they always use normal controls and isolate exosomes from normals to compare with. But for one reason or another, maybe that the balance there between normal and tumoral is different than for cell-free DNA. Because what is quite optimistic about this approach, and these are very recent papers published in 2017 and 2018, it's all preclinical, but 2 studies done in pancreatic cancer and 1 done in non-small cell lung cancer patients. And what those authors claim is that there is a higher mutation detection rate as compared to cell-free DNA. Looking at pancreatic patients with locally advanced disease, and there it was most pronounced, they claim that they found in 80% of the cases or the patients in the exosome-derived DNA tumor-specific mutation, while it's only 30% the case when they look at cell-free DNA in those patients. This was the most pronounced, but looking at patients with very local disease or metastatic disease, there was also a difference. But in their comparison, here it was most pronounced. Looking then at non-small cell lung cancer, also there differences that were observed. They claim 98% with the exosome-derived DNA versus 82% for the cell-free DNA for those activation mutations, and then another particular mutation, which is not telling me so much, but apparently also here was a difference in favor of the exosome-derived DNA. It's all recently, but I think it has potential to look further in, and we looked at it also for our own proposals, and the isolation of the exosomes seems to be a quite easy to perform assay, so I think we have to keep it in mind for the future.